Okay, students, so we'll start the class. There's one important thing and I wanted to put it on record, hence I started the recording. I went through the assignments, and actually the students to whom this is targeted, they're not right now, they're not online. However, um, point number one, I told you all that this was not to be graded, but I don't know why some of you passed on the information to the others who were not interested in attending the class, and they were kind of forced to submit assignments. Uh, I repeatedly said this was a practice session for your term paper and also for your other scientific writing assignments that you might encounter during the course, in fact, your BTech MTech course. But so what I realized was that 4 students submitted the same assignment. And I know who wrote it, but um, he was requested by his friends to send the assignment to them, and very smartly the other 3 changed the name and entry number on the cover page and then submitted it. Okay. So, of course, the gravity of the situation, I don't know how much you understand the gravity, but this is completely unacceptable. Matlab, uh, anything of this sort, whether or not it's graded, is going to be reported to higher authority students. It is the first time that I'm excusing you all. I immediately mailed the students. So at 6:05 or something after the assignment deadline, I mailed the students and I was given some excuses which are lame excuses, because I was told that because it was non-graded and because I was probably just checking the procedure of submission, I don't know what this means, checking the procedure of submission, they submitted their friend's assignment. In fact, one went on to say that it was a joint effort, so they all submitted it. None of this is believable, I know that. I don't understand. If I was checking the process of submission, why would the student then change the name and entry number on the first sheet, or why would the student not submit any random file and just this assignment file? I don't know. But I have taken this very seriously. And students, for you or for any other assignment, I'm not just talking about general microbiology, please be very careful about it. This is not acceptable. You cannot do that. I don't know how many of you have noticed that in my slides, even if I put a cartoon or an animation which is just for sheer attraction or making the slide interesting, you will see a URL down below from where I've taken that picture, okay? Because that's not my intellectual input, or that's not my creativity. I've taken it from somewhere, and hence every slide of mine, almost every slide of mine, has a URL or a reference from which I've taken that particular figure or even text or table. I am not sure how many of you noted it, but please, you can note it from now on. You cannot take anybody else's creation or intellectual, whatever, comprehension or the way they have depicted a thing without referring to it. And मुझे नहीं पता Turnitin का, I did not check whether it was still queued in Turnitin for the assignment, so that I still need to check everybody's assignment. But I was really shocked कि आप लोग first year करने के बाद you do not know what is plagiarism, what is copyright, that you cannot really take somebody else's, और उसमें इतना audacity कि तुम लोग ने change तक नहीं किया, कोई text भी कुछ नहीं change किया, and you simply posted it as your assignment. How can you do that? तो इनफैक्ट आई रिमेम्बर गोपिका ने मुझे पूछा था कि मैम बुलाना है क्या, तुमने पूछा था ना गोपिका?
Yes, ma'am.
एंड आई सेड नो, राइट?
Yeah.
तो फिर तुम लोगों ने क्यों पास ऑन कर दिया जबरदस्ती? तो मुझे बुलाना नहीं है, तुम लोग 6 रहो 8 रहो, आई एम वेरी हैप्पी विद लोअर नंबर्स. हाँ बोलो देवप्रिया.
Some people noticed ना कि वो assignment दिया हुआ था and they started discussing on the group.
तो ये क्या मतलब है यार? मतलब तुम लोग लिख देते कि इट इज नॉट रिक्वायर्ड, तो जबरदस्ती करने के.
मैंने लिख दिया था फिर बाद में कि that नॉन ग्रेडेड है और नेसेसरी नहीं है, बट आई
हाँ,
डोंट
तो उसमें भी आई डोंट नो कि आप सबमिशन चेक कर रहे थे. ये इन्फॉर्मेशन कैसे गई कि हम, क्या मैंने कभी कहा था कि हम सबमिशन प्रोसीडियर चेक कर रहे हैं कि कैसे सबमिशन होता है? मतलब इसमें क्या चेक करना है मुझे? डिड आई एवर से दैट? वन ऑफ यू, कशिश, डिड आई से दैट? रितिका, तुम लोग थे ही नहीं उस दिन क्या?
Yes, ma'am.
Were you there for the assignment? When I put up the assignment, were you there on Teams?
Ma'am, I joined after 15-20 minutes. So at the
हाँ,
last part I was there.
So why did you join? Tell me. After 15-20 minutes, मैंने तो कहा है कोई Teams 5 मिनट के बाद join नहीं करेगा. So why did you join late?
Actually because of some network connections I didn't attend the last 2-3 lectures. So I thought that ये वाली attend करने का sense नहीं है because मैं connect नहीं कर पाऊंगी. But when I got to know कि there is something which you will check, मतलब which you will say कि हमने सही लिखा है कि नहीं. So I thought I should not miss this opportunity and I should hear what you are giving insight about the term paper.
तो पहली बात तो अगर मैंने कहा है कि आप लोग मत पास ऑन करो इन्फॉर्मेशन तो कोई जरूरत नहीं थी बिकॉज आई रियली डोंट वांट टू फोर्स यू टू अटेंड अ पर्टिकुलर क्लास नेटवर्क इशू हो चाहे कुछ भी इशू हो I really don't want you to. This is the reason why attendance is not mandatory. This is the reason why synchronously attending a lecture is also not mandatory. So because we know that there might be issues at your end, even at our end we have issues. बट ये इसका क्या मतलब है कि सबमिशन तो किसी ने तो ये डेफिनेटली मैसेज पास ऑन किया है ना कि आप सबमिशन चेक कर रहे हो कि सबमिशन कैसे करते हैं. I
नहीं
couldn't
मैम, on
even
the
understand.
official group तो this is not shared. As far as I remember, official group पे ये तो नहीं हुआ था कि you are checking कि submit कैसे करते हैं.
तो
It
I
is
don't
just कि
know how
non
this
graded है, हमने practice के लिए दिया था.
किसने किसको क्या पास ऑन किया मुझे पता नहीं है इसलिए डायरेक्ट बात करनी चाहिए और इसीलिए हम अवेलेबल थे. आज तक मुझसे क्वेश्चन I wanted to see कि तुम लोग को ऑनलाइन कोई इश्यूज हो रहे हैं क्या कि ये वर्ड कैसे लिखना है ये टेक्स्ट कैसे लिखना है. बट एनीवे आई वाज रियली शॉक टू सी कि ऐसा करते हो तुम लोग. मुझे पता है तुम में से कोई नहीं है तुम जो अभी ऑनलाइन हो उनमें से कोई भी नहीं है जिसने ऐसा किया है लेकिन क्योंकि ये रिकॉर्डिंग पे है इसलिए आई वांटेड टू पुट दिस आउट फॉर ऑल द अदर स्टूडेंट्स. एंड इस तरह के एक्सक्यूजेस तुम लोग सोचते हो कि टीचर्स लोगों को कुछ पता नहीं है समझ में नहीं आता तो कुछ भी एक्सक्यूज दे दो तो वैसे वाइवा में सब कुछ पता चल ही जाएगा. हाँ तो आई शुड ऑल्सो अनाउंस द टाइमिंग फॉर योर माइनर. It is, I think the deadline is 3 p.m. on the 10th, yeah, 11, on the 11th. So we will start the minor at 1 p.m. on the 11th of November. Okay. क्या ड्यूरेशन होगी क्या इंस्ट्रक्शंस होंगे ये हम आपको बाद में बताएंगे. अभी देयर इज स्टिल टाइम देयर इज अ वीक लेफ्ट सो आई विल लेट यू नो द प्लेटफॉर्म एंड एवरीथिंग बट द स्टार्ट टाइम इज 1. हाँ देवप्रिया.
Can you please repeat the start time? आपकी आवाज कट हो गई थी उस समय.
1 p.m. on the 11th.
Okay.
ठीक है. 3 p.m. is the deadline, so we'll definitely have a. I mean, it won't last for 2 hours, obviously, but we'll start at 1. ठीक है. Okay. I hope everybody is clear with where we stand in terms of the syllabus, with whatever we have covered. What is the syllabus of minor that I've already um, shown you in your syllabus? How much we have covered, and the final 2 topics for minor are number 1, isolation of microorganisms. You've seen how microbes grow. I mean, both the modern mathematical, the growth kinetics as well as the binary fission part. So how individual cells divide and how a group of cells divide. And today we will talk about how to grow them in the lab. So one point that Ashi asked me before we started the class was that in the book there are a number of factors mentioned other than the 4 that we covered. So whether or not that's going to be a part of the syllabus, it's not going to be a part of the syllabus. I took up just some of the factors, though I mentioned pressure and I also mentioned radiation as the other factors which affect growth. But you know that there are certain common or unifying adaptations which are. Ritika, can you please mute yourself? Which are found for every kind of extremophilic condition or every kind of other condition. So I have only considered some of the major ones, the 4 major ones which are also used in classifying bacteria or microorganisms. So whatever I've covered, the topics that I've covered, you have to study those from Prescott. Other than that, you can leave out the chapters in Prescott. Okay, so yeah. It again, I was discussing this before the class that had it been a normal semester, we would have been parallelly doing all these techniques, all these methods, whether it is gram staining or growth or even now cultivation. So, and I don't know how much time we will get when you get to the lab to practice all this. So it's important that you understand the theory behind it, which is also a part of the module of the lectures. So in today's class, we will be talking about different techniques used for cultivation of bacteria. Yes, but before that, tell me, why would you want, in what scenario would you want to isolate a bacteria? Anybody? Isolate a bacteria, matlab? What do you understand by when I say isolate a bacteria? Anybody? अभी तक
Like
इतना समझ हाँ दिव्या tell me.
have a culture where only that particular bacteria is grown.
So why would you need that situation wherein you want just
So
that
maybe
particular bacteria?
study the effects of various reactants on that particular bacteria, or maybe to get the idea about the cell contents by crushing the bacteria and studying it, or or for other reasons like how well does it grow, in what conditions does it grow, etc, etc.
Characterizing it. Let's put
Yeah,
it as characterizing it. ठीक है, चाहे वो physico-chemical characteristics हो, चाहे उसके biological properties हो, that's one thing. Anything else? Why would you need one bacterium in its pure form without other contaminants? What is the necessity? As a biotechnology student, why would you require it? Anybody else? In nature तो नहीं grow करते ना individually. In nature they are always in communities, they are always interacting with other microorganisms. So why else would you require an organism in isolation?
if we want to edit the organism.
Edit the organism. ठीक है, manipulation. Okay, of course you will need one pure culture if you want that to manipulate it or to perform any kind of genetic manipulation. Anybody else? बायोटेक्नोलॉजी के स्टूडेंट्स हो भाई तो बताओ. Characterize करना तो it is more of an academic and research interest, right? To understand a particular microorganism. But then what? Once you've characterized, then अरे एप्लीकेशन नहीं करना है उसका. सारे जगह हो ना. You would require, if you are supposed to, or the intent is to, let's say, produce an antibiotic or let the organism grow in a batch culture for the production of a particular enzyme. Do you want one organism to be inoculated or a mixture of organisms to be inoculated? Why would you want the nutrients to be consumed by organisms which are not desirable? You only want a particular strain to be inoculated so that the particular the target product is being synthesized and the energy is not deviated to other organism, or it's not distributed out to other organisms. So there are various reasons for having a particular strain in isolation. But first, when you say just isolation, what it means is it is the process of obtaining a pure culture. And because you know that in the nature it's a mixed population, so it is obtaining a pure culture by separating one species of a microbe from a mixture of other species. And like you said, why do you need it? The first reason that you said, of course, is that pure culture is required for characterization. And even in our taxonomy class, if you remember, unless you have a bacteria or a microorganism in the pure form, you will not be able to characterize it. Whether it is biochemical or molecular characterization, and even assigning it to a taxon, you needed to have it in your hand, the pure culture in your hand. Then only when you have a pure culture, like I said, for any biotechnological application, whether it is industrial or environment or medical, you need to have a pure culture in your hand. Then why do you know, for some biotechnological application, like for example, you want to start a batch for production of antibiotics, you would like to first check the purity of your starting material. You know that this is the particular yeast of interest, or this is the particular bacteria of interest, and you want to start a batch culture. You would like to check whether or not during preservation the particular microorganism has not got contaminated and is still in the pure form. That is also when you would like to isolate a bacteria and start a batch with a fresh colony or a fresh lineage of cells. And only the desired strain or the isolate is inoculated for the batch process for consistency, for uniformity. Then Cox postulate, agar tumhe yaad ho to, the first step in identifying the causal organism of disease was to isolate it and then re-inoculate it. So that is also important from the medical perspective, that you isolate a bacteria from the mixture, or from, even if it's not a mixture and you want to just check the purity, you need to get one particular colony or start with one particular cell so that there is homogeneity. Now, what is the prerequisite? The flow of our course is such that you first understand the nutritional requirements, then you understand the growth conditions. These are the two prerequisites for our ability to grow a bacteria: the knowledge of the nutrient requirement and the growth conditions. While many microorganisms, they can be grown in a general media. By general media, I mean most of the macro and micronutrients are supplemented. There are some which have a complex requirement. So such organisms, they are called as fastidious microorganisms, or you can relate it to fussy microorganism, fastidious microorganisms, which have very specific requirements. So for them, these organisms, they typically grow and multiply very slowly on normal agar plates, and you require a lot of supplementation, nutritional supplementation. In certain cases, even environmental control has to be checked. So, for example, the best organism that I can cite here is Haemophilus influenzae, which causes influenza. It's commonly found in human respiratory system, leading to respiratory infections. In the laboratory, when you grow this organism, it requires very specific conditions. And in fact, hemolyzed blood is added to the medium for if you want to grow H. influenzae in the lab. So these organisms are called as fastidious organisms. Some definitions that I would like you to know before I actually, because I'm going to use these terms commonly during this PPT. When I say medium, or the plural is media, it is generally a liquid or a gel-like consistency or a matrix designed to support growth of microorganisms. So basically, you have different macro and micro elements and the other growth factors that I talked of as a part of the medium. Inoculum, I think I mentioned to you earlier, this is just the starting material used for a culture, the small amount of material that you use to start culture. This could be anything. If you are isolating microorganisms or growing them from an environmental sample like soil or water or air, it could be that as an inoculum. While it could even be a stored culture in the lab which you just want to grow in a batch, or you want to check its purity, then the inoculum could be just that culture. When I say broth, I'm referring to a liquid medium. Enumeration means determining the number of viable cells. So when you're enumerating, you're trying to count the number of viable cells in a particular sample. Pure culture, like I said, it is the culture containing only one species. And colony, in terms of bacterial growth, is a cluster of bacterial cells formed by asexual division. So you cannot really see individual cells, but if you let the individual cells grow and they form a cluster of cells, you can visualize colonies. But the point here is that a colony, because it is formed by asexual division, should be a homogeneous cluster. It's not that 2 or 3 cells clump together and form a colony, and then it's not a pure culture. One colony here represents a cluster of cells formed by asexual division, so they have to be genetically identical. Then there are different media that are used for isolation and for growth of bacteria. They can be classified on these 3 major parameters. One is the consistency of the media. then the constituents or the composition of the media, and finally, what is the functional property or what is the application of the media. We'll go into each of these and see examples of these three parameters. Consistency means, so like I said, there is a broth or a liquid media which is used for growth of organisms. Now everything has a particular purpose, everything is used for a specific purpose. The second category is solid media, wherein you use a solidifying agent. Commonly we use agar, and this is in approximately 1 to 2% of the medium. And then there's a third type of media, which is called as the semi-solid media, wherein the solidifying agent is lesser. So it's about 1.5% agar that is used. And these media can actually, you can grow organisms in the lab in such flasks, which you will do in your practicals. You will grow them either in flasks or in tubes or even in such plates. Now, how many of you have seen these plates? You know what these plates are called, students? क्या कहते हैं उसको?
Petri dish.
हाँ, right, just petri dish या petri plate कहते हैं, जो कि they are either of glass or of plastic. So this is a liquid medium, as you see, and this is a solid medium. From this this picture, you cannot say this is a semi-solid medium. However, there are various purposes. So we'll talk why when you grow them in tube or when you grow them in a plate even. Now, in liquid medium, so this is, like I said, semi-solid is more like a jelly-like consistency. Okay. In liquid medium, the bacteria, they grow uniformly, leading to turbidity. If the initial number is less, let's say the inoculum has a lesser number than can be observed on a plate or even otherwise, liquid medium is preferred to enhance the numbers. So basically here, the growth is homogeneous throughout this medium. And liquid medium is also used for dilutions. So later you will see why dilutions are important. If you know that you have a pure culture, then you can of course grow them in liquid medium to increase the numbers. Because here you're not able to visualize cells, you're not even able to understand if there is a contamination. So unless and until you're sure that you're using a pure culture, liquid medium does not really help. So here, for example, I know that I have a strain of streptomyces which is needed for the production of an antibiotic. I can grow in a liquid medium to increase the number of cells, but you cannot from this make out if you are growing just the particular pure culture or there's a contamination. So the other medium, that is the solidified medium, which I said wherein the liquid medium is solidified by using one of the solidifying agents like agar. These are used for the isolation of pure culture. Not just for the isolation. तो liquid medium तब था जब या तो inoculum कम हो और आपको increase करना हो numbers, उसके बाद आप उससे isolation भी कर सकते हो on a solid medium. Or if you know that you have a pure culture, then you're growing the numbers by liquid culture. Solid medium में you provide a solid matrix. So the difference with liquid broth is that in this case the cells are fixed, and so the colony morphology can be studied, whereas in the other case you were not sure of what kind of organisms are. If it's a mixed culture, you will not be able to differentiate them. So in the solid medium, you actually provide them you immobilize the cells and let each cell grow on the plate. Initially, Robert Koch used, it is from his time that the Petri plates came into picture. He used gelatin for solidification. In fact, unki assistant thi Julius Petri, who invented the Petri dish named after her. Now, the use of gelatin has certain disadvantages. It is used by many bacteria. So for a solidifying agent, you do not want the solidifying agent to be used up or consumed by the bacteria as a source of nutrient, while gelatin could be used up by many bacteria, which is undesirable because, of course, like I said, you need an inert matrix. And the other problem with gelatin, which was more logistic, was that the gelatin, it is a fluid at temperatures above 28. so which is below the optimum incubation temperature of many bacteria. So this was discontinued, and then agar came into picture. It is very widely used for solidification. It's a complex polysaccharide. The monomers are actually galactose monomers, and it's extracted from a red algae, that's a seaweed. The benefits, of course, it is inert, so no nutritive value to the bacteria. I've put an exclamatory mark. I'll come back to this in a minute. The other important benefit of agar is that it has a broad working range. So it solidifies at about 40 degree and melts only at 100 degrees. So you have a range of working temperature for agar, which was not the case with gelatin. Now, lately it has been realized that some bacteria are inhibited by agar. And there are also reports that talk of agar being utilized by certain bacteria. So again, it defeats the purpose. We are still, in fact, I even saw some reports wherein just a change in the solidifying agent would facilitate some newer cultures to grow. So now that we're talking about just 10% being culturable, there are various groups trying to increase this number. So they found out that even if you change the solidifying agent, you might be able to get a completely different set of microorganisms isolated. So it's not that agar is free of any free of limitations. However, this is still the most commonly used solidifying agent. Now, what is the purpose of using a semi-solid medium? The prime function of this medium is its use in testing the mobility of a bacterium. So the soft consistency, it actually permits motile bacteria to migrate, leading to visualization of turbidity. So if you see in this particular test, the inoculum is stabbed into this the center of the semi-solid agar. This is an agar tube. I mean, the semi-solid medium is in an agar. So a diffuse zone of growth extending outward from the line of inoculation represents bacterial motility. On the other hand, the non-motile bacteria, you will see it grows only in the restricted to the zone of inoculation. So this is a non-motile bacteria. However, this is a motile bacteria. Other than this application, for some bacteria, semi-solid medium is preferred, especially for microaerophiles. It is seen that semi-solid media are better to grow or cultivate them. This was as far as the consistency is concerned. Now coming to the second parameter, which is used for classification of growth medium. Depending upon the composition of the medium could be either a synthetic medium or a complex medium. A synthetic medium or a defined medium, as the name suggests, refers to a medium in which the exact the chemical composition is known, while a complex or an undefined medium is one in which the exact chemical composition of the medium is not known. Now, tell me from the two tables that I have presented, which table is for a defined medium and which one is for complex, or are both of them for complex media or defined medium? Please categorize. Tell me what do you understand from these two tables? Is there anything undefined or everything is defined in the two tables, the left and the right one? Students? बताओ.
Ma'am, left में like you don't know what exactly beef extract contains and in how much quantity. In the right one, you know like the precise quantities of every
How much
compound.
quantity तो लिखा है मैंने amount table
Ma'am,
में
but
दिया हुआ है.
it's given right because it's a source of vitamins and other growth factors. Like, but we don't know how much vitamin is present in 1.5 grams of beef extract.
Yes, so a defined medium has pure chemicals as ingredients, while complex media usually contains some complex materials of biological origin, such as peptone or blood or yeast extract or even beef extract, the exact chemical composition of which is undetermined. So for example, peptone, when you use a peptone, this is actually a controlled enzymatic hydrolysis of animal tissue, and you do not really know of each of the component of it. Similarly, yeast extract is a hydrolysate of yeast, and it serves as a source of nitrogenous compounds, carbon, sulfur, trace nutrients, various vitamins, various growth factors, which are essential for the growth of a diverse group of microorganisms. So this is a complex medium, while this is a defined medium or a synthetic medium. Now, when you use a synthetic medium, you have to know the exact nutritional requirements of the organisms that are being targeted. While complex media, because they provide a range of growth factors that may be required by organisms to, it can be used for organisms for which the exact nutritional requirements are unknown, are still unknown. So complex media are generally used for cultivation of, like I mentioned, the fastidious bacteria, which have a complex requirement. So for them, generally you use a complex medium. Now coming to the third parameter, which is used for classification. This is based on the application of the media or its functional attributes. Basal media, or a general purpose media, again, like the names suggest, it supports most of the non-fastidious bacteria. Some examples, and you'll be using it very commonly, are, for example, nutrient broth, or if it's in the solidified form, solid media, it's called a nutrient agar. These media are generally used for the primary isolation of microorganisms without imparting any bias, because you do not have any prior information as to what is present in a particular sample. So you use a general medium, also called as a supportive medium, to isolate a group of or a broader range of microorganisms. We'll take up all the others as well. In the case of enrichment media, as the name suggests, here you specific groups of microorganisms are enriched, or their growth is stimulated over others. And this is done either by introduction of nutrients or by employing specific environmental conditions which facilitate or promote the growth of the microorganism of interest. So just to give you an example, let's say you want to isolate cellulose degrading bacteria from soil samples. If you add cellulose in the medium, it will selectively enrich the cellulose degraders due to the abundance of these substrates. So similarly, let's say you're interested in isolating a petroleum degrading bacteria or a plastic degrading bacteria. So you add, let's say, crude petroleum or plastic is supplemented to the basal medium or the supportive medium for enrichment. This is as far as the supplementation with a nutrient is concerned. Even the growth conditions can be used so that a particular taxon is benefited. For example, if you incubate your samples at 50 degrees, you are enriching it for thermophiles or thermotolerant strains. So by using the enrichment media, what you do is a small number of the desired organisms are actually increased to detectable limits. The next type, which is called as the selective media, they are used to grow and isolate a specific type of microorganisms, specific type of microorganism. But the difference here is that in the selective media, you suppress the growth of other microorganisms, while in enrichment media, you were not suppressing the growth of the other types, you were just enriching one particular group of interest. Here, for example, the selective media containing antimicrobials or dyes or alcohols, wherein you inhibit the unwanted microorganisms. So for like you add an antibiotic to the medium, all sensitive microorganisms will be killed, and only the medium will be then selective for antibiotic resistant microorganisms. So this is the difference between an enrichment media and a selective media. There is another type of media which is commonly used, and that is called as the differential media. Differential media is used to visually distinguish microorganisms from one another. When microbes are grown in differential media, they produce visible morphological changes or different growth patterns, which help to identify and differentiate one microorganism from the other. Differential media, they are formulated by utilizing the biochemical properties of the microorganisms of interest. And unlike the selective media, differential media are not included with a chemical which suppresses or inhibits. So basically, enrichment selective और differential में ये difference है कि suppression सिर्फ selective media में होता है. Enrichment में one group of microorganisms is enriched. Differential में you can actually visualize the difference between two different sets of organisms. So some media, for example, if you see the EMB medium, which is on the right, this is a differential medium, wherein a color indicator distinguishes between organisms that ferment lactose, for example, E. coli, and those that do not ferment lactose. So a combination of dye, which serves as pH indicator, produces color variation. So this green metallic sheen that you see here is because of the strong acidity because of fermentation of lactose. The non-lactose, so I don't think you can, I'm not sure if you can see the other colonies which are growing here which do not have that sheen. Here is the picture of another medium called as McConkey agar plate, wherein the lactose fermenter E. coli colonies are bright pink in color, whereas the Serratia marcescens colonies, which do not ferment lactose, they form a cream colored streak on the medium. So just by visual observation, you can categorize the functionality of the organism or the kind of organism that you are isolating. Some media which are both selective and differential. So for example, the mannitol salt agar, it is differential because S. aureus or Staphylococcus aureus, it ferments mannitol and turns it yellow. But Staph epidermidis does not ferment mannitol. And if you can see the Micrococcus luteus, this is selective because it is inhibited. It cannot grow on this medium. So while you can differentiate between aureus and epidermidis, this is not growing. So it is selective for only the ones which can ferment mannitol. And because there is an inhibitory substance added, you also call it as a selective medium. Another example is like I showed you McConkey agar. McConkey agar is also selective because it inhibits certain gram-positive like Staph aureus. You cannot see Staph aureus here while E. coli is growing, and you can actually differentiate between E. coli and Salmonella enterica by the color in which you're growing. So if you grow them on a general medium or a supportive medium, there's absolutely no differentiation between the two strains. However, growing them on McConkey, you can very clearly identify between the two different bacteria because of the biochemical differences. This is just to give you an example of what is the inhibitory factor or what is the differentiating agents for different kinds of media which are both S and D. You will see sometimes it could just be a high salt concentration which will inhibit the other kinds of bacteria and promote the halophiles. And like I talked of mannitol salt agar just now, well, there could be some other feature which is differentiating different kinds of bacteria. Now coming to the other type of media, there's a medium called as, or there are various media called as transport media, which are used to maintain the viability of certain organisms in clinical specimens, especially during their transport to the laboratory for testing. So they typically only contain buffers and salts. So if you can see in this example, they're just salts and some buffers. They lack any source of carbon, nitrogen, or any other growth factor. Hence, they do not facilitate microbial multiplication. So you do not want the sample to change from when it has been taken to when it's tested in the laboratory condition. Hence, you call it as the transport medium. Such media, they prevent the drying of a specimen. They preserve the microorganism, and they also inhibit the overgrowth of any unwanted bacteria because you're not supplying anything for growth. And in such media, it has been observed that addition of charcoal. So if you see charcoal is added, it serves to neutralize the inhibitory factors. So if there are organisms which are producing some inhibitory compound, they are just negated by adding charcoal. Some of these media are semi-solid in consistency, and maybe some of you would have encountered such vials in which clinical samples are taken. So basically the samples are taken, let's say, on these swabs, and the swabs are inserted just like I showed you about the stabs, the stabbing method in semi-solid testing for motility. And then they are, because this medium is a transport medium, it will not let it proliferate, but it will keep it viable till it goes to the lab for the testing. Finally, coming to the anaerobic media, these are media meant for anaerobic bacteria, of course. They need special media for growth because they need low oxygen content and reduced oxidation reduction potential, and also some extra nutrients. Media for anaerobes may have to be supplemented with certain growth factors, specifically for anaerobes, like hemin or some vitamins like vitamin K. And these media, they have to be reduced by physical or chemical means. So one such means is to boil the medium which expels any dissolved oxygen and then seal the container. And the chemical means would be, for example, addition of thioglycolate or addition of ascorbic acid, cysteine, or even red hot iron fillings. They can render the medium reduced. One indicator that is commonly used to check for the anaerobic status of the medium is methylene blue, which is an oxidation reduction indicator. And under reduced condition, the methylene blue is colorless. Now, when you grow anaerobes in the lab, the easiest is to cultivate them in a candle jar. It is simply a container in which a candle is lit before sealing it. So the candle's flame burns until it goes off due to oxygen limitation, and then the condition is believed to be anaerobic. So a carbon dioxide rich but an oxygen deficient environment is created in the jar. The other option is using, though we will not be doing any anaerobic culture, the other option, slightly more sophisticated option, is to use gas packs in place of the candle jars, and the gas generator is crushed to produce CO2. This reduces the oxygen concentration in the bag to about 5% and provides CO2 concentration of approximately about 10%. So this jar has the Petri plates that you wish to grow under anaerobic condition. And a still more sophisticated version is if you have an anaerobic chamber that is a sealed anaerobic workspace containing an atmosphere of H2, CO2, and N2. And the anaerobic atmosphere of 0 to 5 ppm of O2 is maintained here by hydrogen gas mix, which reacts with palladium catalyst to remove the excess oxygen. So this is where you put in your hands inside, and these are all sealed. The entire thing is a sealed system. Now, other than using tubes and flasks that I just showed you for growing microorganism, Petri plates are also used. The Petri dishes are also used, which offer a different sort of advantage. Like I said, for a solid medium, the advantage that the Petri plates have is that the cells are fixed, thereby visualizing and counting of cells are possible. The manner in which inoculum is introduced onto these plates. So while you understand that in a liquid medium or in a flask, introduction is easy. The introduction of inoculum is simply adding the inoculum and mixing it or homogenizing it. Here, there are actually 3 ways in which inoculum can be introduced. That is either by streaking, or by spread plating, or by pore plating. And the 3 have very distinct applications. So they serve different purposes. And all the 3 you'll be doing it in your practical classes. A streak plate involves the progressive dilution of an inoculum of bacteria or yeast over the surface of a solidified agar medium, which is in a Petri dish. And the result is that some of the colonies on the plate grow well separated from each other. The aim of this process is to isolate pure cultures. And the technique is also used to check the purity of cultures that are maintained over a long period of time. So regular sampling and streaking is performed. It is also used by certain scientists to start new maintained cultures by picking just one colony of an identifiable species with a sterile loop. So basically, this is the loop which is used for inoculating loop, which is used for streaking. And I'm sure from this picture it is clear. So what you do is you dip it into the inoculum or the starting material, and you start streaking this. The arrows indicate the direction of streaking. So you start streaking the inoculum on the surface of the agar. And once you've done one section, then you take the same loop without dipping it back into the inoculum. You take it from, if this is the direction of your movement, what you're trying to do is the density is going down, and to the extent that towards the end you will have a density wherein most of the cells would have separated. So each cell is actually each colony is actually representing one cell dividing to form a colonies. So the idea here is that you are diluting it such that here then you start picking the colonies or start using the cultures from this section wherein you have diluted it to a cell. There are no clusters formed during this entire process. It's only individual cells been separated out. समझ में आया किसी को स्ट्रीकिंग क्या या कोई डाउट है?
Ma'am, इसमें like why do we
clear है?
hello?
हाँ बोलो.
Ma'am, इसमें
हाँ
like
बोलो.
why do we like make the first few streaks overlap each other like?
Because the inoculum for this streak, this movement is from these that you have already streaked. As you proceed, you are bringing down the density. Okay. तो suppose यहाँ पे तुमने समझ में आया ना, इसीलिए मैंने ये direction दिखाया तुम्हें. Next time you're taking it from this and then this. हाँ. तो progressive dilution करते जा रहे हो तुम. Okay.
Got it.
Fine. So, but here, let's say if you're starting with a sample which has a mixed population, this method does not help you identify or distinguish between morphotypes, because if you see in the first few sectors, you have more of a lawn. You're not able to differentiate between colonies. It's only towards the end that you have a colony. So, अगर आपको एक starter culture, कुछ culture शुरू करना है in a batch, and you have a preserved culture, what you would like to do is before you start the culture, perform a streaking experiment and then inoculate just one of these colonies to be doubly sure that you're starting off with one particular bacteria and it's not a mixed culture. This is the main application of using a streaking method of isolating. But like I said, it has limitations. Here you are not even able to count the number of cells in the first or in the inoculum or in the particular sample you started off with. Because that is not the purpose. In streaking, you're not counting cells. So for that, you need another method which is called as a spread plate method, जिसमें कि the spreaders are of this shape. They're varied shapes. You could have a triangular spreader like this, or you could have a L or a T-shaped spreader, wherein you place the inoculum onto the plate, and then this is a rotating device. So you can rotate and spread the inoculum uniformly across the surface of the Petri plate. And this is just to show you how different dilution. So if you have a higher dilution, you see, you will not be able to differentiate between different colonies. But at a particular appropriate dilution, you can even count as to what was the number of cells in the initial inoculum. Okay. But the point to be noted here is, so here, for example, the dilution is too low. You've got just 2 colonies. Ideally, the thumb rule is that you consider a plate wherein you have anything between 30 to 100 or 300 in some cases, separate colonies. Only then you consider that particular plate for counting. So you see the difference between streaking and spread plate and how the two have different applications. I hope that is clear to you. Here you're trying to count the colonies by spreading the entire sample, and we'll come to the counting now. And while in streaking, the the purpose was to get a pure culture. And here you can even see the diversity with different kinds of colonies found. You can even characterize the diversity of a particular sample. And so like here I'm showing you a combination of liquid culture or broth and the application with spreading. So for example, you have a sample which has a very high density of bacteria. What you do is you serially dilute it for the simple reason that I mentioned that colonies only with 30 to 300 plates only with 30 to 300 colonies are considered for calculating. So what you do is you serially dilute them. This is tenfold dilution if you see, and the colonies wherein you have too many, the plates wherein you have too many colonies are discarded, while you would consider a plate wherein you have, let's say, between that range, here you have 159 colonies, while these again you discard, and you calculate the number of bacteria, you can count the number of viable cells in this by calculating, back calculating kitna dilution factor se kiya, you can calculate the number of cells in the initial sample. Is that clear, students? How serial dilution and plating can help you count the number of cells, and why this method, why can streaking not be used here for counting? Students, anybody? बताइए.
Because there's no uniformity in the dilution, like while streaking.
डाइल्यूशन तो किया ही नहीं था, मतलब
मैम
अच्छा तो प्लेट में बोल रहे हो. हाँ,
हाँ
स्प्रेडिंग ही नहीं हुई है, ठीक है, क्लस्टर्ड है सेल्स अभी यहाँ पे, तो तुम काउंटिंग इनिशियल उसमें तुम कर ही नहीं सकते बिकॉज द एंटायर इनोकुलम हियर इज ऑल लॉन्ड, इट्स अ लॉन्ड, देयर इज नो सेपरेशन ऑफ सेल्स, राइट. तो यहाँ ये पर्पज ही नहीं है काउंटिंग. इफ यू आर आस्क्ड टू काउंट द सेल्स यू हैव टू गो फॉर अ मेथड लाइक स्प्रेड प्लेटिंग. ओके, and just talking about the final method, then we'll stop. Pour plate is the third method which is used. It does not require plate preparation in advance. The pour plate technique can be used to determine, this is also a method used for determination of the abundance of microbes in a specimen. What is done here is the sample or the inoculum is pipetted into a sterile plate and then you add the medium. This can also happen by you adding the inoculum onto the flask, in the flask which has the molten agar, and then you form the plates and incubate. So basically this is for homogenization of a smaller volume of inoculum that you have in a larger volume of medium, such that there is uniformity in the cells. And once you incubate them, you find colonies like this. There could be surface colonies, there could also be subsurface colonies, because you've mixed the inoculum into the medium, unlike the streaking or the spreading wherein the colonies are all surface colonies. तो वहां disadvantage ये था कि only the aerobes can grow by streaking and by spreading unless you keep them in an anaerobic chamber. While here, because they are subsurface colonies, you can also see that microaerophils and I wouldn't say anaerobe, but microaerophils definitely can grow under these subsurface conditions. So, but this is a time-consuming method. If you're not looking for microaerophils, you would prefer to go for spreading. And the other disadvantage here is because you're adding the inoculum with the molten agar, which is above 40 degree, you might be introducing a bias in killing the microorganisms which are not mesophilic, because of the heat shock that you might encounter or the cells might encounter. But here, like I said, the advantage is uniform mixing and also some of the permitting the growth of some of the microaerophils. Another logistic issue here is when you have subsurface colonies, unlike the spread plate where the visualization is quite easy, here counting becomes a little difficult. When you would like to count the number of cells, it's a little difficult here. So yeah, I hope the three techniques of plating are clear to you. I just wanted to show you this picture of colonies of yeast, such beautiful colonies of yeast formed by streaking. So you see the lawns here and then individual colonies towards the end of the streak. and now it's a very common event. Agar art competitions are very common. This is quite, it's not as simple as it looks like. So with all the information that you might have, it's not just the color of the cells that's important, it's also how the organisms grow respective to each other, that you get a picture like this. These are very common in microbiology conferences, and I've just put in some reference material for you all to go through. If there are any questions, please ask me, students, about the methods that we've just talked of. किसी को कोई प्रॉब्लम? ये सब चीजें आपको प्रैक्टिकल में करनी है, तो उस समय थ्योरी नहीं बताया जाएगा, इस समय सारी चीजें थ्योरी में आपको क्लियर की गई हैं. ऋतुराज, तुम्हें कुछ क्लियर हुआ क्या आइसोलेशन के मेथड्स? बिकॉज आई नो यू विल बी यूजिंग दीज मेथड्स.
यस मैम. एक्चुअली मैम आई हैव अ डाउट इन स्प्रेड प्लेटिंग. तो मैम जो स्प्रेड प्लेटिंग में जिस टाइम पे हम डाइल्यूट करते हैं, अगर हम एक सेल बता रहे हैं कि एक सेल कंसंट्रेशन है, तो एक कोई एक पर्टिकुलर रेंज है जिस पे हमें बोलना होता है कि मींस वेयर वी हैव टू स्टॉप कि इतने नंबर ऑफ जो सेल्स है, या फिर इतने नंबर ऑफ काउंटिंग के लिए जो है इतने नंबर ऑफ सेल्स होने चाहिए उस प्लेट में. एक्चुअली इन वन ऑफ द ड्राइंग
हाँ, मुझे समझ में नहीं आया, लेकिन आई ट्राई टू आंसर. सो बेसिकली तुम जब रिप्रेजेंट करते हो ना, तो यू से CFU और कॉलोनी फॉर्मिंग यूनिट पर ml ऑफ डैश. ठीक है. when you back calculate, let's say you're starting from soil sample, 5 gram soil से तुमने शुरू किया, इसका एक slurry बनाया because तुम्हें inoculate करना है, so you make a slurry of add let's say 10 ml of water. उसका तुमने dilution बनाया because you do not know the abundance, right? So you cannot simply plate 100 microliter को तुमने plate नहीं करोगे. तुम उसके dilutions, initially तुम्हें dilutions बनाने होंगे, उसके बाद तुम calculate कर सकते हो. 10 पर 3 dilution पे आया तुम्हारा काउंटेबल नंबर ऑफ कॉलोनीज, देन यू कैन. तो मेरी जो सीरियल डाइल्यूशन वाली स्लाइड थी वो इसीलिए थी कि आप बैक कैलकुलेट कैसे कर सकते हो. ठीक है. बट यू नेवर से इट इज द नंबर ऑफ सेल्स, यू से कॉलोनी फॉर्मिंग यूनिट, बिकॉज व्हाट यू आर काउंटिंग आर कॉलोनीज. यू अज्यूम दैट ईच कॉलोनी इज फॉर्म्ड बाय अ सिंगल सेल. ठीक है.
यस मैम.
एनीबॉडी एल्स हैज अ डाउट? अदरवाइज वी विल वाइंड अप देन.
And ma'am, one more question, ma'am. Whenever we transport a soil from a place, so is there any time range in which we have to transport it to the laboratory, or
there is no ideal transport. से भी ज़्यादा important है where you store it, how you transport it. है ना? When you're talking of, what I was talking of was the transport media for pure culture, but what you're talking of is transport of an environmental sample. So what you try to do is to bring it as close as possible. So avoid any fluctuations, whether it is temperature fluctuation or humidity or whatever. But what we prefer is to, in fact, the next class we will talk about preservation of cultures. The best is to inhibit or bring down the microbial activity of the particular sample, so that you are storing it or preserving it in a state which is closest to what you sampled it. Okay. But we will touch this topic in the next 2 lectures. Okay. Anybody else? Okay, fine. Then if there are no more questions, we'll stop the class. We will see each other now on Thursday then, students.
Thank you.